When you want to apply your model at large scale, it's very important to talk to the people, to involve the people that work with a large scale system, because they know they have very interesting knowledge that you need, and it would be wrong to ignore them. So I think there's a human factor there because they know some things about specific operational issues which could be important for you to know when you try to validate your model. So it's important to work on such a modeling exercise. For example, transferring from small scale to large scale. It's not just an individual effort. I think it's a team effort.
Welcome to the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we're diving into the fascinating world of bioreactor modeling and online sensors with Krist Gernaey, who is a professor in Industrial Fermentation Technology at the Department of Chemical and Biochemical Engineering at the Technical University of Denmark. As an expert in industrial fermentation technology, Krist brings invaluable insights on scaling bioprocesses with confidence. If you're wrestling with bioreactor variability or scale-up challenges, you are about to discover game-changing strategies to streamline your development process. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Krist. It's good to have you on today.
Yeah, thank you. Also very nice to be here finally.
Yeah, it's a pleasure, Krist. It has taken some time, but as we said in the briefing, good things take time. So I'm really looking forward to our conversation, Krist. Krist, share something that you believe about bioprocess development that most people disagree with.
Yeah, well, I don't know if people disagree with what I will say, but I am convinced that if you look at the bioprocess development and bioprocess operation, I am convinced that if you spend a little more effort to develop or to work with better measurements, so to have more informative data, then this pays off because you can then work with simple control algorithms to be able to run your process. That's what I'm convinced of.
Yes, isn't it all about data and actually using them well and have the tools to have the right accuracy and precision, right?
Yeah, having tools to convert data to information. Because having lots of data does not necessarily mean that you know something about your process.
And this is exactly why we're here today, Smart Biotech Scientists. We're going to talk about data. We're going to talk about process optimization, using data the best way possible, and a lot more. But before we do that, Krist, draw us into your story. Tell us about what sparked your initial interest in bioprocessing and what professional journey took you to the work that you're doing today.
It's complicated, as they say. 25 years ago, nobody thought that I would go this direction because I've done my PhD on developing measurement technologies for wastewater treatment systems. And at the end of my PhD, there came an interest in modeling. And then I spent most of my time as a postdoc to work on modeling of wastewater treatment systems. And then around 2005, so about 20 years ago, I was so lucky to get a position here at DTU as associate professor. And then my field of interest changed from wastewater treatment to biotechnology. And basically, I was asked to develop projects that go in the direction of biotechnology. And what I was really interested in all this bioprocess technology was the fact that it's a very rich field. I mean, there are so many different products, so many different organisms. And of course, thinking back about my PhD and about the postdoc work I've done, I was mostly interested to work with measurement technologies, measurement techniques, and then also try to work with mathematical models applied to bioprocesses. And then I've been lucky to be able to attract some funding already early on. We managed to hire some great PhD candidates with some excellent collaborations. And then things just accelerated. And at some point, then I was appointed as a full professor and have since then also developed a lot of collaboration with different industrial partners, because that's also one of the aspects that I enjoy a lot about working in the field where I work. That is to see that some of the things we develop also have value at full scale, not just in the lab or at pilot scale.
Krist, what do you see as some of the main challenges today in industrial fermentation technology?
One of the main challenges nowadays is that there's a lot of talk about digitalization and people talk about advanced things like digital twins. But I think there is a big gap still between what is actually implemented and what people talk about, because the implementation itself shows to be much more complicated than people expect. It's just challenging to go from an environment where not so much is available online to move to an environment where data are available in the cloud, where everything is suddenly available, where you can work with data and then just find ways to make good use of that. I also think that one of the challenges at large scale is that the set of measurement data that you have available is rather limited. And people have to learn to accept this, because it's no use to develop a very advanced control strategy which relies on some exotic measurements which works in the lab. Beforehand, it will be very difficult to transfer this to full scale. And I think this is something that people need to understand when they start on such a development journey.
You're making a good point. You have to have the scale up or the end. You have to have the end in mind right from the beginning, because if you use some fancy stuff that works in a lab, it doesn't mean that it works well in the large scale, or it can actually even be very complicated to use that in large scale.
Yeah, I agree. And of course, I do not say that you should not start working with new technology, but when you start developing new technology, you always have to be aware that the conditions at full scale are quite harsh and are very different from what you can do, what you experience at lab scale. So people need to be aware of that. And of course, another thing which is different comparing lab scale to full scale is that at full scale, cost becomes a very important factor. So preferably if you develop some measurements, or if you want to work with some measurements that you want to use in the frame of a control strategy, well, preferably you should use some measurements which are affordable, because then the chance that they get implemented at larger scale is also increasing a lot.
And I think what is important when you're developing new technologies is that you have an awareness of the requirements of large scale. And when I was working in technology development many years ago, there was a time when it was good to be creative and to look at new technologies in the lab. But then we had this moment to say, okay, what do we now need to do in order to transfer that into production and to sell it also to those teams? Because all of a sudden you have some pushbacks from those guys. So I'm now wondering also about the general state of the industry. You said in the data field, the majority is probably not there where it could be yet. But overall, the biotech industry or the fermentation industry, do you think we are at maturity, or it's still ways to go there?
I think it will still take some time to get there. But I think there is a growing awareness of the fact that by looking at data, you can actually create some additional value. And just comparing when I started working in this field, let's say 20 years ago now, and nowadays, there's much more discussion about using models, using data in a production context compared to 20 years ago. I think we should accept that this is also the time it takes to change things. I mean, if you look at a typical production, the bioreactor is there for many years. And if you want to change something, it just takes time. So if we can accept that, then maybe we can drive some change.
Now let's talk about the modeling, because you mentioned data, you mentioned modeling, you mentioned the digital twin. We're going to talk about the digital twin a bit later. But first of all, let's talk about the modeling itself, because there are different modeling approaches. Give us an overview of the different modeling approaches we have at our disposals, and what are the pros and cons of each approach for different applications?
Originally, at the start of my research career, let's say, I was only focused on working with first principles models, so mechanistic models. The nice thing, of course, with these type of models is that you can extrapolate also through conditions where you did not really collect any data. The disadvantage is that it takes time to set up these models. So these are models which are based, for example, on mass balances or component balances. Then you add the relevant kinetic phenomena and so on. I mean, it takes time to set up these models, and it can also be pretty complicated to validate these models, for example, when you start applying them at larger scale. Then completely the opposite end of the spectrum, we have data-driven models. So where you only look at trying to connect relations between data using different types of algorithms. Yeah, that's, for example, say neural networks. To give one example, could also be simple regressions, could be simple multivariate approaches. But basically there you have the advantage that setting up a model is relatively simple. But the disadvantage is that the amount of knowledge that you create by setting up a data-driven model is relatively limited, and there is challenges with extrapolation outside the range of data you have used to calibrate or to estimate your model parameters. I think the past years especially, there has been quite some focus on hybrid models. So a hybrid model is that a model that combines a mechanistic part with a data-driven part. And that I see as a very strong combination because it allows you to use, let's say, the easy parts from the mechanistic world. So, for example, a mass balance, a component balance. And then in terms of kinetics, you could, for example, try to update your kinetics based on the data that you get from your system using a data-driven model component. For example, estimating a growth rate, estimating a production rate. That I think is very powerful. And it also helps to compensate for some of the disadvantages of purely mechanistic models, for example, in fermentation, where typically the yields in an unstructured model are often constant. And in practice, we see that especially for longer fermentation, the yields are changing a bit. This you could compensate by working in a hybrid modeling approach.
And now let's say you have developed a hybrid model in the lab, you have done some testing, some runs and so on, and now comes the time to think about an industrial application. So what methodologies are you using, and what would you advise to a biotech scientist who is asking, well, should I use hybrid modeling, and if, how should I use it?
Yeah, that's actually a difficult question to give a good answer to, but I think that you should, as much as possible, when you work with hybrid models, already take into account when you develop them at smaller scale, that the data set you will experience at larger scale will be limited. When you apply these hybrid models at larger scale, the most challenging is to find good ways to validate that your model predictions also hold at larger scale, which is not necessarily the case because at larger scale, we typically struggle with the fact that we have gradients in our bioreactors. And this means that it can be important to choose a proper measurement point because depending on where you measure, the data that you will collect might be slightly different or might even highlight some different phenomena. So I think the most important attitude you need to have is to be critical when you transfer a model, any model, not just a hybrid model, from smaller scale to larger scale, and to take care that you validate properly at larger scale that the model also performs well there. And I think in general, model validation at large scale is one of the challenges we have because to properly validate a model, you also need to get to collect an extensive data set to be able to do that. And that's typically very time consuming. And that's, I think, an effort which many people underestimate.
To what extent can you reduce the validation effort at large scale? Because now many companies they validated a small scale system to do, for instance, the process validation and so on. What is your experience there? Do you usually still do validations at larger scale? Or now with, let's say, with machine learning algorithms or new language models, can you actually fully skip that?
I think it's dangerous to skip that, but you could, of course, try to use the tools that you have to help you in trying to validate the models. You could, for example, consider not to collect experimental data at large scale. You could also consider to try to generate them using another model. Just give an example. If you are so lucky that you have a computational fluid dynamics model of your large-scale system, then in principle, you could reduce it to a compartment model and then use the compartment model to generate some data which will resemble rather closely what would happen at larger scale. You can combine this with tools like Monte Carlo simulation to be able to create some variability in your data set. And in this way, maybe create something which could at least give an indication if your model is also performing well at larger scale. Because experimental work at larger scale is very challenging.
Yeah, it's challenging and it's expensive. So what I'm hearing, it comes down also to acquiring a lot of process understanding in the small scale, actually tend to know what you still need to do in order to fully validate your model.
I think there's also a human aspect that it's very important when you, as a scientist or as a model, when you want to apply your model at large scale, it's very important to talk to the people, to involve the people that work with a large scale system, because they know they have very interesting knowledge that you need, and it would be wrong to ignore them. So I think there's a human factor there because they know some things about specific operational issues which could be important for you to know when you try to validate your model. So it's important to work on such a modeling exercise. For example, transferring from small scale to large scale. It's not just an individual effort. I think it's a team effort.
Yes, absolutely. It's a team effort. And when we speak about modeling, we should also speak actually what happens up front, because we need excellent data in order to create a powerful model. If the data is not collected the right way, or it's not accurate, or whatever issue it might have, your model will not be good. So let's talk about first the way actually we should gather this data, because you're working on new online sensors and for fermentation. So tell us a bit why you're working on this and what are the current limitations. And then tell us also actually what is the most effective way to implement, for instance, a real-time control then with this to make the best use out of the data.
Traditionally, when we talk about large scale, I'm used to a situation where you have standard measurements available like pH, dissolved oxygen and the like. But if you want to have information about, for example, substrate concentrations, product concentrations, then these are typically measured offline based on samples which are taken from a bioreactor. And that is, of course, not frequently enough to be able to use it as input to real-time control. What you then can consider is you could try to estimate, you could try to work with algorithms that can allow you to estimate, for example, a product concentration or substrate concentration. But these approaches, I think, are in many cases rather complicated. So I would rather prefer to try to measure these things directly. The challenge is that if you look at a fermentation broth, it's a very challenging matrix to measure in because you have a combination of different types of particles. It could be particles that originate from the substrate, so from the feed. It could be the biomass particles. Sometimes it's individual cells. It can be clumps of cells that you have filamentous organisms. It becomes even more complicated. Then you have, in aerobic fermentation, you have gas bubbles that interfere with your measurement. And then in general, you have complex media. You have organisms that produce all kinds of components. So especially, I think, if we talk about spectroscopy, it's very complicated to work in a fermentation environment, in a fermentation broth. That's why what we have tried to do the past years is try to develop electrochemical measurements which try to quantify individual components. And this works reasonably well at lab scale. The challenge is now how to expand this to larger scale, because you need to make the measurement such that it's robust enough to survive in a large-scale fermenter if you want to measure data. So the dream is to be able to obtain, for example, electrochemical measurement that can tell you, okay, glucose concentration or sugar concentration is evolving like this. Your product concentration is evolving like this. And then you could use this directly as input to a control algorithm.
And how does this physically work? Because do I have to imagine that we have to have a sensor for each attribute? For instance, one for glucose, one for lactate, one for another? Or is that one single sensor that does everything?
We work now on individual sensors for each component, but the dream is to be able to combine them in a single probe. So, for example, have a device that looks like a pH probe, but can actually measure, for example, glucose, lactate, ammonium for you. And what is also very important if you talk about measurements is to not only be able to provide proper data, to provide high quality data, it should also be a measurement which has a simple calibration procedure. So what we aim at is to develop measurement devices which have which work with 2-point calibration. So it's similar like pH, for example. Because my experience with spectroscopic measurements applied to fermentation is also that the calibration procedures are very tedious, very time consuming, and therefore also very expensive.
So that's how you would differentiate your new electrochemical methods. So for instance, the standard probes the industry is using, right?
But as said, we are not there yet. We are still developing. We have some positive results at lab scale. We are now trying to expand this to a system which could be applied at larger scale, at full scale.
That wraps up part 1 of our insightful conversation with Krist Gernaey. we've explored the evolution of bioprocess modeling and the critical challenges in industrial fermentation technology. Struggling with your own bioprocess scale-up challenges? Or are you wondering to implement better sensor technology in your facility? I'd love to help you tackle these questions directly. If this is of interest, book a free call with me at BrühlmannConsulting/call. Stay tuned for part 2, where we will dive deeper into digital twins and the future of bioprocessing with AI. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.